First PUC biology, one of the important chapter is biomolecule. So biomolecules chapter, some of the important topics are here. So the molecules present in living organisms are called as biomolecules, and it includes carbohydrates, fats, proteins, nucleic acids, vitamins, etc. And except lipid, all the biomolecules can form polymers, and lipid cannot form a polymer. One of the 3 marks question is the type of bonds present between the monomers in a polymer, or the type of nature of bonds in biomolecules. One of the important bond is peptide bond. It is present between two amino acids in a protein. And phosphodiester bond is a bond present in nucleic acid between two nucleosides. And glycosidic bond present in polysaccharides between two monosaccharides. So for 3 marks about the nature of bonds, peptide bond, phosphodiester bond, glycosidic bond, they are going to ask. 1 mark question, starch is a storage polysaccharide in plants and it can form color with iodine. And glycogen is a storage polysaccharide in animals, so it is called as animal starch, important for 1 mark. There are two types of amino acids based on the nutrition, important for 2 marks. Essential amino acids we need to take through the diet, and non-essential amino acids they can be prepared by our own body. So for 2 marks this is very important. For 1 mark, collagen is the most abundant protein in animal world, and rubisco is the most abundant protein in plants and in whole biosphere. Rubisco is the most abundant protein in whole biosphere, whereas collagen is the most abundant protein in animals world. Then protein, very important for 4 or 5 marks. Protein can exhibit in 4 level of organization, or about the structure of protein they are going to ask. The 4 levels of organization of protein are primary structure, secondary structure, tertiary structure, and quaternary structure. So about this, very important to the exam. Now about enzyme and its classification, important for 5 marks. So this is a 5 marks question. Enzymes are proteinaceous substance. They are called as biocatalysts because they catalyze the chemical reaction. Now there are 6 classes of enzyme. Classification is very important to the exam. Enzyme classification. Oxidoreductases, they catalyze oxidation reduction reaction. Transferases, they catalyze the transfer of functional group. Hydrolysis, catalyze the hydrolysis of different types of bonds. Lyases removes the group from the substrate. Isomerase convert the isomers into another isomer, and ligases catalyze the linking together of two compounds. So classification of enzyme is very important to the exam for 5 marks. Then for 1 mark and 2 marks, they'll ask the definition of cofactor. Cofactors are the non-protein part of the enzyme. They make enzyme more catalytically active. For 1 mark, they'll ask the definition of apoenzyme, and apoenzyme is a protein portion of the enzyme. And another 1 mark question is ribozymes. So ribozymes are the nucleic acids, especially RNA, which act as an enzyme. They are called as ribozyme. So another 3 marks question is enzyme action. In enzyme action, we can see enzyme react with substrate to form enzyme substrate complex, and that enzyme substrate complex break down to give enzyme and product. Enzyme substrate complex break down to produce product and release the enzyme for the next catalytic action. So this is very important for 3 marks. Now for 1 or 2 marks question, they may ask the definition of primary metabolites and secondary metabolites with example. So primary metabolites, they are found in animal tissue. They are very important for normal physiology. Example, amino acids, carbohydrates, proteins and all. Secondary metabolite definition is still very important for 2 marks. So they are the metabolites. They are found in plants and they are very useful for human welfare. Example, rubber, essential oils, antibiotics, pigments, gums, spices. Secondary metabolite is very important to the exam for 2 marks. Next, the factors affecting the enzyme. So there are many factors that affects the enzyme activity. So first one is substrate concentration. If you increase the concentration of the substrate, enzyme activity also increases. pH. So every enzyme has an optimum pH. So below that optimum pH and above the optimum pH, the enzyme get denatured. About temperature also. Temperature is also very important factor. So every enzyme has a optimal temperature for its maximum activity. So any alteration in the temperature leads to the denaturation of enzyme. So cofactor and coenzyme also plays a very important role in enzyme activity. So they increase the catalytic activity. And last one is enzyme inhibition. There are some enzyme inhibitors. They bind to the enzyme active site and decreases the activity of enzyme. So they are called as enzyme inhibitors. So these are some of the important topics related to this chapter.